سلام ریز لهمو لگشت گوگرین هیجا ایرو 25مین ریز سمینار زیلمو پیش کشو دکنه میوانه مای هیجا ایرو پروفسور دکتر مظهر عدلیه کجا با کردستان هاتیه و جای شانازی شونه شانازی سامرا تایبتی یک پرجی سامرا های ایرو که ما مستا نفر یکومه کجا با کردستان بوی میوانه ما لزیلمو و امجی سپاسیوی دکنه دکتر ادلی پروفسورا زانکو نورث مدیکال سکول کو یک جا زانکوی هر جور دنیا اگر تماشا بکن سالین پر او یک جا دهه دنیا کو دیسا جی سامر شانازی ساگل کرد را به تایبت دکتر ادلی لمجارا جنتیکس یا ژنتیک و ژنتیکا مروان کار دکه از گرگا من بو که دکتر ادلی به ناس کرم بهم بوه اما خوی دکتر ادلی کرم کریه چند اسلاید اک سامر چه کریه کجا که هاتیه چون هاتیه گشته به امریکا و چند اسلاید پیشیه که دکتر بومه به سامر رو به کرمانجی دیجه به زاراوی کرمانجی کردی دیجه جای وقتی که تره نام مجارا ساینس یا علمیه سامر به انگلیسی پرسه سمینارا ما مستجی 40 دقیقه بعد از سمینار 20 دقیقه سوالان پرس بکن سوالان جی به هر زاراوی کردی که حزدکن پرس بکن یا به انگلیسی و از با ما مستا ترجمه دکم و ما مستجی به انگلیسی ساینس جواب برسو ده بوم ما مستا کرم کم لخدمتت
سپاس دکم مصطفى گلی گداران هر گلی حوالا اونگش بخیر هاتنه ایرو جوارا ام چه دکم وی قصه دکم و سلایدا آنی براک از کیمه هندکی ورا بحث دکم دکترا خال امریکا گرت ل یونیورسیتی اف نورث کارولاینا چاپل هیل گرت دویرا چون باستن م پست دکترا خال ودر کرد آنکای جاز ل Northwestern University سیدامه اما ازندکی دخوازم که پیشوی قصه بکنم اندکی از خلکی ارزورمه از لارزورمه بوم دویرا از چوم آنقره و اسرا دو یک سالی از چوم آنقره جامعه پیخوام ملوی داری خواندن دا از استانبول دویرا هاتم امریکای جبوی دکترا خواه از باستن م پست دکترا خواه از صفحه بوم پروفسور شارلزویل یونیورسیتی اف ویرجینیا شارلزویل اما از سالدن از وای سالی که از هاتم شیکاگوی از طبیعی لازرمی بوم لهندری شهری ارزرمی دانه بوم اوا گنده من از لوی داری لوی گنده بوم گند کی پر قجی که 20 مال هنه یانتنن 100 مرو 200 مرو آنجا خیلی و مالا ما والوی داره و طبعا گندی آنکای گندی بلی اینها بینای اینها تنون یعنی بلای اینها چاتی تنون بهار افا پر خواشه طبعا اما زمستان خواهی پر زوره یه ویدرا پر برفت باره یعنی وسترز بوم لگند من دو کلت نبود از گند کی نزدیک مهو 2 کیلومتر لما بدات رو امو چند تنه حوالی خواهم دوام لیده شنو گندی دنت هاتن دورا دورتا وكذا از شم آنقره مل آنقره خواند و دین طبیعی و این حیات منو لبی در بازو اما از ایرو دخوازم که قصه بکم جبوی حیات ما جبوی حیات و دنیای گشتی که قصه بکم انا کاز در باز کی انگلیسی دکم مالسف بقى کرمانجی بلیم قصه نکریه یعنی so now I'm going to switch to the English part. So I want to talk about life in general, not only my life, my small life, but the big life. As you know, it has been almost 4 billion years that life has been appearing on Earth. but with the one major question that we all ask: what drives life? What drives all the beautiful forms of life? And the answer: there are two main forces that drives life. One is every life forms have DNA in them, and the one major form that drives this life is random mutation, and the second form is just natural selection. And those two processes, they drive life on Earth. We, as our species, as humans, we have changed this a little bit since the last 10,000 years or so. The mutations were still random, but we start to non-naturally select things around us. You can think that 90% of species around you, if you look around you, every tree you see, every animal you see around you, every bird almost you see around you, we humans, we selected them. None of them are probably in their natural forms. This, of course, gave us immense power to be able to modify the environment around us. That's why we used to be one of the weakest species on Earth, but now we are one of the strongest. And probably right now we are becoming the strongest species on Earth that is shaping everything around us. But now we have a completely new tool around us. It is much more powerful, more powerful than we ever imagined. It is so powerful that now we don't need to wait for random mutations to occur. Now we can non-randomly introduce specific mutations in our DNA and also in the DNA of other species around us. Now we can change their genetic information. And of course, this speeds up our effect on this world to unprecedented levels. So now we can make non-random mutations, and we can continue to non-naturally select these things. And the main tool that's enabling us to do this is the tool that we call CRISPR. Well, of course, CRISPR is the newest tool, but it's not the only tool. What does it allows us to do is that it allows us to this genetic information that every living thing has this in their code. This is the code of every living organisms, and it used to be read-only file. We could not make any changes on this file. We could read it, and it only happened in the last 70, 80 years or so that we could gradually able to read this file, but we were not able to make any changes. And CRISPR is basically allowing us to not only read this file, but also edit this file, make any specific changes. And the word specific here is very critical. It's not that we are randomly making some changes in the DNA. We are able to very precisely change this genetic information. As I mentioned, the CRISPR is the latest form of this quest to edit DNA, to change DNA. But the adventure of trying to understand DNA didn't start just recently. So ever since we knew that DNA is a genetic information, the things that passes from fathers and mothers to their next generation, ever since we understand that this is DNA in 1940s, and the DNA structure was resolved in 1952, the first papers. It got the Nobel Prize, of course. Then we try to understand how we can change this information. And that's why all the restriction enzymes that got Nobel Prize, these are specific proteins that recognize specific DNA sequence that will cut DNA. And it's enabled us to kind of like together different DNA pieces to understand their function. The first, of course, we could do this one in test tubes, but we wanted to do this in living things. So the first attempt to change genetic information in living things was this knocking in specific DNA sequences in mouse. This was a huge game changer. Now we could change things in living things, but it was a very, very inefficient process. Only very specific labs could do it, but still it was a big step forward, and this got the Nobel Prize as well. Then comes the targeted genome engineering. So The first attempts were, meganucleases, zinc finger nucleases, TALENs, and now the most recent CRISPR that got recent, the Nobel Prize. All of this new targeted genome engineering, their goal is to make a double-strand break in DNA. So here is a more schematic version of these things. I don't want to go into the details of all of these tools, but the first 3 tools were protein-based, and it was very difficult to engineer a protein. But this tool, this new CRISPR-Cas9, it is a combination of protein and RNA. Here, we can change around 20 nucleotide, and then this enables us to custom target any specific DNA we want. So for the audiences who may not know the difference between protein and RNA, RNA is a nucleic acid, proteins are amino acids, and it's very easy to synthesize nucleic acids. We are able to fuse different nucleotides to each other and make any custom RNA we want. But proteins are much more difficult to engineer. All of these guys, what they're trying to do is make a double break. Now, you may imagine, why would we want to make a double-strand break? Because DNA is the most essential molecule for any living organism. The answer actually is also within this information that since DNA is such an essential molecule, throughout evolution, organisms develop different types of mechanism to cope with this DNA damage. Because this is a damage, basically. You are damaging DNA. And organisms have to repair this, because if they don't, they may lose one big chunk of DNA. And of course, this is not something that any organisms want. So there are 2 main mechanisms that living things use to repair this. One we call non-homology and quick fusion of these broken ends to each other. And during this process, random mutations happens. So for an organism rather than losing a big chunk of DNA, they move on with just having few mutations here. This is a very quick and efficient version of DNA repair. Another alternative repair is that, since every living things, higher organisms at least, they have 2 alleles, 2 templates basically. So an organism can use the other non-broken template to repair this. But we as a human species, we can introduce new templates as well. So by introducing new templates at this broken site, we can introduce, for example, a new gene, or we can introduce a new mutation. So this is another way we can make a very precise gene editing. In this case, we are just deleting genes. So this is very easy to just delete a gene. This is very good to introduce a gene. And this one is much more efficient. Non-homology end joining is much more efficient. However, the homology-directed repair is very rare. But we have developed sophisticated ways to select those cells that has this kind of repair. So, but what is CRISPR now? So CRISPR is a bacterial immune system, actually. So this was discovered a long time ago, that in bacteria has this weird sequences that kind of repeats. So this was in 1987, this was first discovered. But for almost 30 years, we had no idea why bacteria should have these repeat sequences. The answer kind of appeared in around 2005 when computational genomics people figured that some of these sequences that are in between these repeat sequences, they don't belong to bacteria, they belong to viruses. First, immediately, all scientists thought that if a bacteria is carrying a piece of virus within its own genome, it must evolutionarily provide some sort of advantage to bacteria. And the only answer that comes to our mind is using as an immune system to recognize that virus again. And the answer was correct, that bacteria, by containing some part of virus in its own genome, kind of carries the picture of its enemy within its own pocket. That's why if that virus infects the bacteria again, the bacteria can use this CRISPR to recognize this virus and chop it off its DNA. system in around 2012, scientists figured out what is the minimal component of this system to work, and whether we can transfer this system to another species, to human cells and other things. And that's what the major breakthrough happened in 2012, that we discovered the minimal components of this system, and we could also transfer this system from bacteria to human cells and show that it also works in human cells. So the system worked with the Cas9 as a protein, and then with a short piece of RNA that we can easily synthesize, and we can target anywhere we want this protein with this short RNA, around 20 nucleotides. It will specifically bind to the target sites, and it will cut it. This is what we call wild-type Cas9 is doing. But we can also make specific mutations in Cas9, and we can make it inactive. And so you may imagine, why would you want to use this one? The answer is that we can use this inactive version, but we can fuse different effector proteins to this one. We can fuse different enzymes to this Cas9. This will enable us to specifically target Cas9 to a specific region in the genome, and we can make other specific functions. For example, we can add unique epigenetic modifications. We can remove epigenetic modifications. We can turn genes on and off, or we can image genes. My lab, the research in my lab, is focused on understanding genome. And we want to understand how genome is differentially regulated in normal settings as well as in disease settings. When we talk about genome is the whole genetic information that any organism has, the entire genetic information. Of course, we, like multiplex organisms, multicellular organism, we have this information in every single cell. And in our case, in human genome, we have around 3.2 billion letters, AGCT for information. And we have around 20,000 genes and more than 1 million regulatory elements in our genome. When we say genome regulation, we can think genome regulation in at least 3 separate layers of regulation. The first layer, which, when we immediately think about DNA, we always think about this naked DNA. This just AGCT next to each other. But this is the primary DNA sequence, but DNA never is like this in a living organism. It is always packed with other proteins that we call chromatin. So the main protein component of this organization are proteins we call histone proteins that form this bead on a string structure. But even this is not the ultimate regulation. What happens is that depending on higher order structure, these things can be packed in different parts of the nucleus in 3-dimensional space within the nucleus. For example, we know that if a gene is highly active, it tends to stay in the center of the nucleus. If it's inactive, if that cell is not using that gene very often, then those genes will be packed on the periphery of the nucleus. So we try to understand all these different layers of regulation. And of course, CRISPR just became the main tool for us to be able to do this. Now, with using CRISPR, we can change the genetic information. We can also change chromatin structure. We can visualize 3D organization and all this. So this is some of the ways people have been using CRISPR. So we have normal Cas9. We can do gene editing. We have this base editing that is also highly, to me, it's very exciting new field. We can make specific nucleotide changes. We can turn A into G. We can turn G into C without making double strand break. So this is a new form of CRISPR in a way. We can do epigenetic editing, and these are some of the papers that my lab has published in this related field about the normal Cas9, about base editing. We could do epigenetic editing with using this inactive Cas9, but fuse different epigenetic modifiers. This way we can deposit new epigenetic information, we can remove epigenetic information, and this enables us to turn genes on and off. So this may something easy to do right now, but a few years back, this was a holy grail. We were always imagining whether we would be able to one day turn genes on and off. Imagine we have a tiny nucleus, and within that tiny nucleus, we have 3.2 billion letters of information and 20-something thousand genes, but we want to go and turn only one gene, turn it off. And this was not an easy thing to achieve. But now CRISPR enables us to do this kind of things. We could do topology editing. We could do chromatin imaging. We can actually take videos of individual genes, and we can, in a live cells, we can visualize where this gene is, how it behaves over time, for example. So, and then there's also a CRISPR system that targets RNAs. So CRISPR used to be just wild type and inactive, and new functions are being added to this CRISPR technology. And now it's becoming this really multifunctional tool that you can do many things with this CRISPR. So the research in my lab, has a couple of kind of research goals in parallel. One is technology development. So we use CRISPR-based tools to develop new technologies if we need to. The two major focus of the lab is to understand chemoresistant in ovarian cancer and identify synthetic lethal drug combinations for cancer. So it's a cancer-focused lab, cancer functional genomics. The other way is to keep using these tools to understand genome organization in normal settings as well. And one part of it is more personal, is I'm also interested in CRISPR ethics, and I will mention a little bit about this, that if we have this powerful tool, there are a lot of ethical implications of this as well. So this synthetic lethality, so I want to talk about this synthetic lethality a little bit, and what does it mean in terms of cancer. So synthetic lethality term is coming from yeast genetics. When scientists were deleting one gene, the yeast is viable. And when they delete another gene, it's still viable. But there were some gene combinations, when they were deleted together, the yeast was not viable anymore. So the idea now inspired the cancer researchers that in cancer, of course, we cannot delete genes because it's much more complex, and we have to delete this in every single cancer cell, which is impossible. So instead, we use drugs. So the idea is that we add drug A, the cancer cells are viable. They don't change much. Maybe they respond a little bit. We add drug B, they respond a little bit. But we hope to find these two drug combinations that when together, the cancer cells are not viable anymore. Of course, in reality, we want to achieve synergism here. The synergism means that, let's say if drug A is killing 10%, drug B is killing 10%, the combination, we want to achieve 80%, not just 20%. And that's what we mean by synthetic lethality, what we mean by synergistic effect. And of course, we want to have the synergistic cytotoxicity. The other thing we want is that these two drugs will selectively be toxic to cancer cells, not normal cells. That's another essential part of this. How do we achieve this? We want to target into pancreatic cancer. So this is just a very brief information about pancreatic cancer. Pancreas is actually two glands kind of fused together. On one hand, we have these exocrine cells, which secretes all the digestive enzymes, and 98% of the cells in pancreas are these exocrine cells. We also have these endocrine cells, these beta cells that release insulin and glucagon. And they only occur, they are composed of only 2% of the pancreatic cancer cells. So pancreatic cancer, which is 95% of them are from these exocrine cells. They are very dense cancer, so it's very lethal. 80% of the patients die within the first year. So it's like a death sentence. In most cancers, there is a great improvements in terms of statistics, in terms of the percent patients who survive. But in pancreatic cancer, we unfortunately, the statistics are still very bad. Although we know the genomics of it, we know which mutations happen in pancreatic cancer. Like KRAS is almost universally in every pancreatic cancer, KRAS oncogenic mutations, and that's the mutation in these tumor suppressors. But we are not able to target any of these. So because of these challenges, it is predicted to be the second most lethal cancer by 2030. And we wanted to do CRISPR screening to figure out what drug combinations we can use for pancreatic cancer. So what does CRISPR screening means? So this is a super powerful application of the CRISPR technology. So I mentioned that we can use a single short RNA to delete a gene in any cells we want almost. But imagine a situation where instead of just using one single RNA, we can use thousands and thousands of RNAs and deliver this with the virus to a population of cells. So in this case, in a millions of cells, in each cell we are deleting different gene, okay? But every cell is kind of barcoded because we deliver with the virus, and this enables us to integrate this barcode into the genome of every cell. And we deliver at a rate that each cell will receive one virus. So this kind of enables us to barcode every single cell and at the same time knock out or delete a different gene in each of these cells. And then we apply some sort of selection. This can be just time, this can be a drug. And we, for example, in this case, this green cells, after, let's say, a period of, there are a lot more green cells in the population than other cells. This suggests that when we delete these green genes, the cells even proliferated much faster. So this will be potentially a tumor suppressor gene, for example, a gene that normally blocks cell cycle proliferation. And now when we remove that gene, the cells proliferated much faster. So we wanted to, of course, do the screening for combinatorial drugs. The goal is to take a pancreatic tumor cells, deliver this library, and then do the in vivo CRISPR screening, because in vivo, our physiology, the cancer physiology behaves differently in vivo versus in culture conditions. And we inject these cells into the pancreas of mice, allow the tumors to form. And after a while, we are currently, for this particular project, we are interested in these red genes. So these red genes are that in the normal control, when we delete these genes, tumors didn't care much. But when we add drug, in this situation, gemcitabine, which is a main chemotherapy that is being used for pancreatic cancer patients, when we add this gemcitabine, these red cells, they disappeared from the tumor, indicating that by themselves, just deleting PRMT5, for example, in this case, the cells were surviving fine. But when we delete PRMT5 and add the drug, then they couldn't survive. I won't go into all the details of verifying this information, but this is just the final experiment that we did to show that this could be potentially a new therapeutic target for pancreatic cancer. So here in this case we are injecting in the same mice. We are on one side, we are injecting a wild type tumor. The other case, we are injecting the tumors that don't have this gene. We call PRMT5, it's an enzyme. So normal tumors, both of them form big tumors. But when we add drug, only the knockout tumors shrink, the other one don't shrink. This showing that this could be a potentially new combinatorial target. Of course, in cancer, we cannot delete genes, as I said, we have to use drugs. And this gene happens to be one of the druggable genes. So here is that experiment. Instead of deleting it, we are using a chemical to inhibit this gene, this PRMT5. And as you can see, a single agent, they barely have any effect, but the combination of them, they kind of didn't allow tumors to grow. And now we are excited about this result. So we are in negotiations with SmithKline to start a clinical trial here at Northwestern using this combination. So I mentioned about this powerful CRISPR system, but when we have something so powerful in our hand, we typically come across with a lot of ethical considerations and ethical implication of such things. The easiest one is, are we going to now genetically modify humans? Are we going to have a designer babies that we can define their height, their eye color, their skin color, their IQ levels? But we realize that this is actually, we are getting so, and actually recently when this CRISPR came out, I wrote this article about this ethical implications of this one. And just 3 months later, a Chinese scientist modified embryos with CRISPR, and now CRISPR babies are living and are born. I hope they are still alive. But this immediately tells us that we are at that age. We can actually change genetic information in our species as well as many other species around us. And CRISPR is giving us such power that we never had for many, many thousands of years. And now we are at that age that we can actually design this genetics in living things. And I was interviewed, although I'm not a medical ethicist, but since this publication happened and this thing was born at the same time, a lot of news articles and news outlets, including Chinese national TV, we interviewed with me about this ethical considerations after these CRISPR babies. So this became one of my kind of side personal venture to read and think and more about these ethical things. So now that we are able to do it, what does it mean to edit humans, to change the genetic information in humans? Well, there are two types of cells that we can change in humans. One cell is we call somatic cells, which is 99% of our body is composed of the somatic cells. If you change anything in the somatic cells, let's say in our skin cells, in our blood, in our whatever cells, most of the cells, if you change this information, it may affect this particular individual, but it will not be passed to the next generations. So this is the category of gene editing. But if you change anything in the germ cells, in the sperms or eggs, then this will change to the next generation and next generation and next generation. So this part of the human genome editing, there are a lot more concerns with this part than this part. Okay. So the current guidelines, the current thing is that we should not change genetic information in germ cells. And of course, if you do it in embryos, then the germ cells will also be modified. So the current, kind of gentleman's agreement is that we should never do germline editing. When a rogue scientist comes up, like the one that Dr. He in China, he does it, then all of the world and all of the scientists, they're like, what the heck, what happened now? So we thought we are not going to do it, but there is no regulatory entity that can regulate this kind of things, unfortunately. So about somatic editing, there are different ways to do somatic editing, and there are a lot of diseases that we can cure this way as well. There are 6,000 monogenic diseases, and we can potentially use CRISPR to correct most of these genetic diseases. Of course, there are a lot of challenges in front of us, including the delivery of CRISPR system to organs, etc. But technically, in theory, CRISPR can cure all these genetic diseases. Cure it. So there are different ways to apply CRISPR to such things. So generally, we think CRISPR should be used for curing diseases on the somatic cells, not on the germ cells, okay? We can do in vivo gene editing, meaning that we can package this CRISPR and deliver to the organ, the target organ, systematically maybe. Just to target the organ that is most affected. There are some certain organs that you don't have to correct 100% of the cells. In fact, diseases, you don't need to correct 100% of the cells to get rid of symptoms. For example, cystic fibrosis, the organ that is most affected is lung. And although 100% of the cystic fibrosis patients don't have this specific gene that is required, but if you correct only 10% of the cells in lung, you get rid of 90% of the symptoms. So that's why it is quite powerful to do this one. But for some other diseases, it's going to be more challenging, like muscular dystrophies, for example, where you cannot just systematically deliver CRISPR so that every muscle will be corrected. You have to locally inject it, and this, you can imagine how challenging this could be. Some other genetic diseases, mainly hematopoietic or blood-related malignancies, we can take patients' stem cells, we can isolate the DNA, isolate the blood stem cells from them. we can ex vivo, outside the body, we can do genetic editing and give these cells back to patient. And now there are a lot of therapies already approved for these purposes. Were, as a species, human species, we were one of the weakest species on the face of Earth. But now we are dominating everything, and we are using and exploiting all the animals, all the plants for our own purposes. Of course, the big philosophical and ethical question is, who gives us this power? Why should we be so powerful on this planet? Out a species on the space. We can technically right now get rid of mosquitoes in the face of Earth. You can imagine why would we do that? Because there are some diseases that is transmitted from mosquitoes in Africa and other parts of the world, and it's every year it is killing 750,000 humans. And technically, we can just get rid of those mosquitoes. The way it works is that this CRISPR system, you modify some mosquitoes in the lab. You modify it in a way that when this mosquito is mating with the next generation, all the next generation is going to be male or going to be female. And after a while, every member of that species are going to be male or going to be female, which will not reproduce anymore. So the whole species collapses. And you can imagine that this can potentially cure a lot of diseases for humans, malaria type of diseases. But the things that you are tackling, the thing that you are playing with is such a destructive thing. What will happen to the global system now, global ecosystem? How are we going to control this? What are we going to do if this thing gets out of control? These are all the ethical things that we are talking about. And there is, for our human thing, we talk about humanized animals, for example. We are talking about growing organs in different species, in pigs, in other species. And now with the help of CRISPR, we can even do this completely and humanize animals so that our body won't reject those organs. But the ethical side is that this chimeric animals, basically, you are growing something human in some animals. And there is a lot of questions about animal welfare. Why should we have this control over animals? And there are these other ethical things as well. For our case of human applications, I mentioned that somatic genome editing is thought that it can be done as long as it is treating a disease. And germline embryo editing, we should not do it. But if someone does it, how are we going to control this? Now, for the somatic editing, I mentioned that as long as it cures a disease, the current understanding, the current agreement is that we can do it. But what is a disease and what is a normal? So the boundaries there are not very clear a lot of times. For example, for someone that has a muscular dystrophy, his muscles are wasting every day, it is a disease and we should cure it. But at the same time, someone can say that, well, I want my muscles to be even bigger. I want my child or my soldiers to have even much bigger muscles. And right now, it is so easy to just change the muscle mass in any species right now, because we know which gene is controlling this. Myostatin is a gene, for example. If you delete that gene, the muscle mass it doubles almost. And there are a lot of natural form of mutations in this gene in a lot of different animals. For example, blue Belgian bull, for example, it is this huge muscly bull, and the only reason why it is so huge is because of the mutation, natural mutation in that gene in that species. And it happened, I don't know how many thousand years ago, but that bull species is super muscular. The same thing happens in a lot of sheep or goats, for example. They are naturally have this mutation in this gene. And if you do this mutation in humans, 99%, I guarantee that probably there won't be a lot of side effects, and this will be a super muscular human. Now, should we do this one? Of course, there's a huge ethical things. Even for somatic cell editing, that you cure a disease, there is still some ethical questions there. For example, is this tool going to be available for everyone, regardless of their economic status? Or is it going to be just used by some rich people about this thing? And this enhancement versus cure is a big dilemma. Let's say cholesterol. If cholesterol is super high, it is considered as a disease. So we can reduce the cholesterol level. But someone says that I want my kids to have even much lower cholesterol, much healthier even. Now, should we do this kind of things? So there is a lot of ethical implications for this tools and technologies. So with this, I would like to actually thank you for listening, and I would like to thank the people in my lab who has done all the work. Although I didn't talk about much data, but this work, the lab has been run by people in the lab who developed a lot of these things. And as I said, we moved to Chicago last year. It's a beautiful city, and we look forward to have new members in our lab, especially computational biologists. We are looking forward to have new talents in the lab. And with this, I would like to again thank you for listening, and I will be happy to answer any questions that you may have. And I think, yes, we are not too much.
Thank you very much. Yes, خوش ماموستا زور سپاس هم به انگلیسی اجم هم به کردی زور باش بود دست خوش اگر کسی پرسیار اکایه بیجن هم لخصمت
مصطفى من دتوانم
خواهش دارم بفرم نخوازی هر به انگلیسی پرسکه که کریم اره
Thanks very much, Kak Mazhar. This was a great talk. As we know, Kak Mazhar, that the CRISPR is still in development, and there are many different types of CRISPR every day that people are working on from different other species. I mean, mainly bacterial, and we know also that there is also lots of off targets, and it's not that 100% efficient to target just the gene that we are interested. And also we have lots of limitation due to the repeated regions in the genomes that makes also off target. With all of this, and also the delivery challenges, that the way that we should be able to efficiently deliver mainly to the somatic cells. And together, all of these challenges that we have, of course, it's a very nice and powerful tool for maybe at the moment in agriculture and also for basic research studies. But for human genome modification with all these challenges, it's a big risk. Of course, China is something else that we know. They do things there that people in Europe and in the US, they are not allowed. But what is your perspective, I mean, your future prediction about this tool in the near future? Do you think that it's become in the clinic in 5 years, in 10 years, or what?
Yeah, that's a great question. I think there are a couple of smaller questions within your question. So one is, are there off targets of CRISPR? We want to change one thing in the genome. Does it change some other things in the genome as well? So the short answer is that CRISPR has off-target effects. In fact, this was the first article that we published in our lab, that CRISPR has off-target effects. We used CRISPR chip-seq technology to map all the binding sites of CRISPR. and we found that CRISPR does have off-target effects. So now, this was, of course, almost 8 years ago. The CRISPR has been improved tremendously since then. So 10 years ago, if we say there are 5, 10 off-target effects, now probably CRISPR has 1 or 2 off-target effects. Maybe in the near future, it will be none. So the beauty of it is that we will know, and we know exactly where these off-targets are. So once we do an experiment, once we design a guide RNA, we can actually, within our technical power, of course, which is nowadays is pretty amazing actually, we can know where else we are modifying in the genome. And we can map these off-target effects. Of course, if it is mapping one of the important genes, we cannot use it for therapeutic purposes. We cannot use that guide RNA. We can design a new one. So we can fine-tune these things until we get a perfect CRISPR system. Now, so that it doesn't have off-target effects. This is one aspect of it. But the CRISPR, as I said, has been improved tremendously. Now, let's put the CRISPR aside. What does it mean to have some off-target effects? Of course, if you are talking about embryo editing, we should not do it. And even if one day we start to do it, we have to make sure that we don't have any off-targets and that it's safe. But for therapeutic applications, having some off-targets doesn't mean much. Let me explain myself. You know, we are using chemotherapy to treat cancer, to treat many other diseases, right? This chemotherapy, the main way that it works, it makes tons and tons of mutations randomly in the body. Talk about, for example, gemcitabine, talk about cisplatin, any DNA damaging agents, which is most of the therapeutic chemotherapy agents, make tons of mutations, but we still dump this in body and we try to do it. CRISPR, if you just think about in terms of random mutations, CRISPR is at least 10 and 10,000 times much more precise than all this chemotherapy. Of course, still, CRISPR is not perfect. It has some off-target effects. But it is being improved tremendously right now. So the second part of your question is whether we will see, you know, these things in the near future. What are the near future implications? I personally think that, you know, our species, you know, we and our generation are going to witness all sorts of genetic modifications. For therapeutic implications, FDA has already approved some CRISPR for some local diseases, let's say macular degeneration, CAR T systems, cancer immune therapy. CRISPR has been already approved for this kind of things. But I think, you know, it is going to be a form of enhancement, and we are going to witness this one, not only for you know, curing diseases, but for enhancement. And we, as our species, we love enhancing ourselves, you know, and it will be a time probably not CRISPRing your children will be seen as something bad. If you have a power, probably our society will demand that you should do it. Why not? You know, if you can, why not? Like this dilemma, whether this is going to be available to only rich people or poor people, I think will remain there. But this dilemma is there for everything else as well, right? Rich people, they get a special tutor for their kids. They send their kids to a private school. They enhance their kids throughout their life. And poor people are not able to do these things. And I think CRISPR will have something similar, that rich people will be able to modify it, whether it's a cholesterol, whether it's a muscle, whether it's other things. But it's probably not going to be in the next 10 years. But our generation, I think, is going to witness this thing.
Yeah.
Any other question?
Thank you. And I have one other question, but maybe if someone else has a question, first take that.
Yeah.
I have a question about gene drive later.
Okay. Anybody else?
من برسيارك ما كاي مصطفى. Thank you very much, Dr. Mazhar. That was really great. So I have a question about the way CRISPR was discovered. I'm curious to know whether these repeat elements that you mentioned that exist in the bacterial genome, is it really just a simple repeat of just one same virus, and it is different between different bacteria, or bacteria basically carry hundreds of thousands of pieces from different viruses that they get infected with?
So it's a great question. So within the same CRISPR array, there are parts of this array that's repeats in between these spacers. There are parts of the DNA that belongs to different viruses. It's not all the same virus that bacteria keeps integrating. But evolutionarily, it integrates the virus that is presently, growing with this bacteria at the same time. So whichever virus species was around that bacteria for the longest time, it contains the array for that particular virus. And CRISPR system changes tremendously from one bacterial species to another bacterial species. You're right. The current form of CRISPR that we widely use is from this bacteria, Staphylococcus aureus, that we use this CRISPR system. But there are alternative CRISPR systems in different bacteria. And some CRISPR just targets DNA. Some CRISPR systems just target RNA, for example. It's entirely different. And the proteins, they all more or less have similar kind of structure, that there is a protein and there is an RNA that helps this protein to guides this protein. But the shape of proteins, the catalytic subunits, the size of proteins are completely different from one species to another.
Thank you very much.
Any other question? Mazhar, if you allow me, I also have a question. I was
of course
thinking about the GMO when you mentioned that if we consider it as a GMO or not, because I'm working with this stuff also. There was also, when we've been passing our undergrad courses, there was a conventional breeding and mother breeding and gene manipulation, and there was all of those arguments. I think that same thing happened for the CRISPR kind of, there is always a discussion about that. But my question is, how, you said there is some, basically they allow to do some gene modifications with CRISPR in US. What is the procedure, like for the plants, for example, that I know, there is some tests, some steps they have to pass, lots of stuff like that to be considered, like they allow that plant. In US, we have at least 10 GMO plants that they basically we use them. I'm just wondering, there is like a proper organization, proper protocol for doing that? Do you know about that?
Yeah, for plants, I don't know honestly the details of this regulatory organization, but as you know, in the US, everything has to go through several steps. So there are a lot of regulatory process on that end. The current kind of dilemma or debate is, whether you should label this as a GMO or not. That's a big thing because producers don't want to label it as GMO. They think it's not a GMO. The reason is that GMO has this negative connotation with it, and the public acceptance of GMO has not been great. Although as a scientist, I know that GMO has no deleterious effect. There are some other problems with the GMO, that you change the ecosystem and now the GMO, your plant is now resistant to a chemical, and you dump that chemical to kill everything else. And that chemical is going to make a lot of other damages to the environment, to the ecosystem. But the fact that you have a piece of DNA in that plant doesn't make that plant any worse than the wild type plant. But I don't know the exact regulatory process for plants in America.
And for the animals, like GMO animals, we don't have any publicly available GMO animals yet. We have lots of plants, but not animals. I was just surprised that they allow to do for disease and
so,
yeah.
For animals, we have salmon. We have a GMO salmon that basically has this gene and it is produced right now. Okay. It becomes a much bigger salmon, and also it it develops much faster. So it kind of, I don't know the exact the time frame, developmental time frame, but if the wild type is, let's say, takes 3 years to fully develop, the modified one is 1.5 years. So they cut almost 50% the developmental timing, and it becomes much more muscular. So the salmon is being sold right now. The other thing that is being used in terms of experimental applications, for example, in cows, there's dehorning. You know, it's actually a very inhumane process that farmers, they don't want their cows to have horns, big horns, because they damage each other, and also they damage the farmers. So they want to cut their horns, and this process is very inhumane. You know, literally with the sow, you cut it. It's very bloody. With the CRISPR, we know which gene is controlling horn formation, and we can modify it, and now all the animals would be no horns, for example. Again, there's a lot of applications in pigs, and there are companies who only doing CRISPR pigs for various different purposes, mainly for this humanized, pigs, to grow different organs in pigs. There are these companies who are actively right now doing it, but they haven't sold as a product yet.
Mm-hmm.
Thank you very much. Thanks for everybody to watching us and listening to us. زور سپاس کاک مظهر، سپاس دکم جهمو حواله هجا
سپاس دکم
و دارم که سربرزو که خوش بونو کاک مظهر اگر اخوازی تشتکتر اضافه بکی بیجه دعای سکت هرچی اخوازی بیجه.
I would like to thank you guys for giving me this opportunity. Diako, Mustafa, thank you for translating. Look forward to meet you guys in some meetings.
Sure. هر بجین زور سپاس مالا باید کم با همون حواله هجا روژ شب